So, I have to continue about the physiological roles of gibberellic acid. GA basically regulates several physiological processes such as bolting, genetic dwarfism, increases seed germination, break seed dormancy, promote fruit set, floral initiation and sex determination. Now we discuss about the process of bolting. The bolting is characterized by the elongation of the shoot or stem that is significantly influenced by the application of gibberellic acid in plants subjected to short days. Further, under normal conditions, endogenous GA plays a regulatory role in the natural bolting process. GA also play an important role in seed germination, breaking dormancy, promotion of fruit set, and it enhances the seed germination by increasing the activation of hydrolytic enzyme. During the germination process, the seed relies on stored reserves such as starch, proteins and lipids. GA acts as a key regulator in signaling pathway that trigger the synthesis and activation of hydrolytic enzymes. These hydrolytic enzymes including amylases, proteases and lipases are responsible for breaking down complex storage molecules into simpler form. Coming to next physiological role, that is breaking of dormancy, birds produced during winter often enter a state of dormancy, a period where growth and development are temporarily suspended due to the prevailing low temperature. GA has proven to be a potent agent in breaking this dormancy. GA treatment effectively triggers the initiation of growth processes within dormant birds. The application of GA serves as a signaling mechanism, overcoming the inhibitory effects of low temperature and stimulating the resumption of physiological activity. Gibberellins play a crucial role in the promotion of fruit set, which marks the initiation of fruit growth following successful pollination. Interestingly, application of gibberellins can induce fruit set and stimulate the growth of certain fruits, particularly in a situation where auxin, that is another plant hormone, may not have exert a significant effect. One such example where GA stimulated fruit set was observed in the case of apple. Gibberellin demonstrated the capacity to substitute for the environmental cues such as long days or cold condition that many plants, particularly rosette species, typically require for floral initiation. In these instances, GA functions as crucial component of the flowering stimulus, playing a key role in the induction of the flowering processes. The ability of gibberellin to replace the necessity for a specific day length or cold temperature underscore its regulatory influence on the genetic and physiological pathways associated with floral initiation. By acting as a floral stimulus, gibberellin promotes the transition from vegetative growth to the reproductive phase in plants, ultimately leading to the development of flower. Moving to next important physiological role regulated by gibberellic acid is sex determination. In males, the application of exogenous gibberellic acid to the tassels have been observed to induce the development of pistillate flowers. This suggests that GA plays a role in sex determination in males, influencing the differentiation of female reproductive structures. Similarly, in crops such as cucumber, hemp and spinach, the application of GA has been found to promote the formation of staminate flowers. This implies that gibberellin has the ability to influence the development of male reproductive structures in these plant species. The commercial application of gibberellins. A major use of gibberellin is to increase the stalk length of seedless grape. Because of the shortness of the individual fruit stalks, bunches of seedless grapes are too compact and the growth of the berry is restricted. These commercial application highlights the diverse roles of gibberellin in agriculture, horticulture and the food industry, contributing to improved crop productivity and quality. So now I will discuss several other commercial applications. First one is fruit production. As you can look in the image that gibberellic acid stimulates the elongation of stalk in grape vines. This elongation allows grapes to grow larger by reducing compaction and promoting the expansion of fruit clusters. The application of gibberellin is a horticulture practice aimed at improving the overall size and quality of grape bunches. A combination of benzyladenine, a cytokinin, along with GA4 and GA7 gibberellin has been found to induce the elongation of apple fruits. This mixture is specifically used to enhance the shape of delicious type apples under certain conditions. The cytokinin and gibberellin combination promotes cell division and elongation, contributing to the desired elongated and well-formed appearance of the apple. In the malting process of barley, gibberellin is employed to enhance specific aspects. It plays a crucial role in breaking down of starches within barley grains, promoting the activity of enzymes like amylases, which hydrolyze starch into fermentable sugars, a vital step for subsequent fermentation in brewing. Additionally, gibberellin's application ensures a more uniform germination of barley, contributing to standardized malt quality and overall process efficiency. Furthermore, the GA facilitates faster germination, a valuable trait in commercial malting operation where rapid turnaround times are essential. while the use of gibberellins varies and is contingent on brewing practices and malt characteristics. Its occasional implementation underscores its potential to optimize the malting processes, particularly within brewing industries. Spraying sugarcane crop with gibberellin has demonstrated the potential to significantly boost the yield crop. So, the application of gibberellin, a plant growth regulator, contributes to enhance raw cane production and increase sugar yields per acre. Gibberellic acid's application results in a notable increase in raw cane yield with reported improvements of up to 20 tons per acre. In addition to elevating raw cane production, GA also influences sugar yield. GA enhances the accumulation of sucrose in the sugarcane stalk, leading to an increased sugar yield of approximately 2 tons per acre. This is crucial for sugarcane farmers and the sugar industry as it directly affects the economic output and profitability of sugarcane cultivation. GA plays important as well as potent role in plant breeding. GA is employed to stimulate the development of male flowers in cucurbits. In biennial rosette crops like beets and cabbage, gibberellins are utilized to induce bolting. As I have discussed earlier, bolting refers to the rapid elongation of the stem that leads to the initiation of flowering. Now, let us discuss about the gibberellin mediated signaling pathway. The process unfolds through a series of well-defined steps. Initially, GA1 from the embryos binds to a cell surface receptor. Subsequently, the cell surface GA receptor complex engages with a heterotrimeric G protein, instigating two distinct signals transduction chain. Following this, it involves a calcium independent pathway wherein cGMP activates a signaling intermediate. Then the activated signaling intermediate binds to delta repressor protein in the nucleus. Following this, the degradation of delta repressors when bound to the GA signal, thereby inactivating them. This leads to another step where the inactivation of delta repressor permits the expression of the MYB gene along with other genes to progress through transcription, processing and translation. Moving to another step, the newly synthesized MYB protein enters the nucleus and binds to the promoter gene for alpha amylase and other hydrolytic enzymes. Consequently, the transcription of alpha amylase and other hydrolytic genes is activated. Then, this alpha amylase and other hydrolases are synthesized on the rough endoplasmic reticulum and secreted via the Golgi apparatus. Finally, the secretory pathway is facilitated through GA stimulation involving a calcium calmodulin dependent signal transduction pathway. This sequential cascade of events highlights the intricate molecular mechanism underlying gibberellin induced processes, particularly in the context of gene expression and enzyme synthesis. As we wrap up today's discussion on the GA, I want to express my gratitude for your engagement. Thank you for being a part of our session.